Again, if you didn't get 100%, I did reopen your worksheet so you can give that another whirl. The first part prepares you for today's quiz, going over divisions of the nervous system. The second part, kind of a review of material covered in the screencast yesterday. Yesterday, you did cells of the nervous system, neuroglia, and neurons. I actually meant to put more of the actual cells on that piece versus the anatomy of the neuron. Again, I reopened the worksheet for those of us who didn't get 100%. As we go through this, you want to maybe take note of your errors. So we started our day with the Wiser Me worksheet. We'll go over the answers together, and then we'll take the nervous system quiz. There are only 10 questions, so it should take us less than 10 minutes. Multiple choice. And then physiology of the nervous system. A lot of it should sound very familiar from the muscular system. And then we'll have a follow-up worksheet that goes with that. Hopefully you find your worksheets have been coinciding with the lectures this week. So let's look at our interactive worksheets. The first section was the divisions. Central nervous system, we're going to go all the way down here to the brain and spinal cord. Peripheral nervous system, following that all the way down here to the nerves. So the brain and spinal cord versus the nerves that extend outward. Then we have the opposites, sensory versus motor. Sensory is sending impulses to the CNS. Remember, we also call that the afferent division, A, arrive. The motor division, down here to sending impulses away from the CNS. Remember, we also call that efferent, E for exit. And then the next pair of comparisons, somatic versus autonomic. So somatic goes to the skeletal muscles, voluntary. The autonomic goes to smooth muscles, glands, and organs, typically involuntary. Our next pair, sympathetic versus parasympathetic. Sympathetic, we identify with fight or flight, emergency situations, mostly you're speeding things up. And parasympathetic is associated with rest and digest. Non-emergency situations, usually you're slowing things down. Any questions on the divisions part? Okay, then you have two types of cells, neuroglia and neurons. Neuroglia are the supporting cells. They provide protection, insulation, anchoring, things like that. The neurons are the ones that you probably think of when you think of nerves. They are the ones that are sending signals. So they can conduct electrical impulses. Structure of a neuron, it has a cell body, also called the soma. The soma is where you find the nucleus as well as other organelles. They have two extensions. Dendrites go toward the cell body. These are our receptors, the receiving signals. The axon goes away from, goes away from the cell body. So these take signals to other cells. The axon is covered by a myelin sheath that protects it as well as speeds up the neural transmission. And then finally, at the end of the axon, you have the axon terminal. Terminal means end. This is where you connect with synapses, release neurotransmitters like acetylcholine into the synaptic cleft. And that is the anatomy, basically, of the neuron. Questions over this worksheet? Hopefully you were able to get in and make changes for any mistakes you might have made. So I think I will go back to our agenda slide. So the next thing we have is our quiz over the nervous system. So I need everybody's cameras on. You are going to need your Chromebook. So if you don't have your Chromebook nearby, maybe run down and get it. Like I said, it's just 10 questions. So we're going to be moving on again in 10 minutes. I think multiple choice is really just like 30-second questions, but I'll give you a minute for each of those. So I have it in our Google Classroom. One moment, I have to hit assign yet. It's called, ooh, it said it's 50 points. It's not, it's only 10. Ungrade, oops. Miss, look. So actually, let me look at my friends, see if you're all ready to go. I got cameras on. I'm looking at more than foreheads. How about Kirsten, were you having problems with your camera last time I talked to you? Is that right? Brother, is sister in the house?
No, I'm sorry. I wasn't.
Oh, you do have a camera. Okay. I couldn't remember. I remember somebody last time did. Okay. So great. You guys all look so good. I love it when you turn your cameras on. Okay, I'm going to hit assign and you will need to just one moment refresh your screen in order for that to show up in your assignments. Lots of good scores. All right, so that was 10 points, but I'm making it out of 9 because I think a lot of people forget the afferent, efferent divisions are the sensory and the motor. So I hope I have more than 5 people, but if not, at least you 5 people are here, and that's what's important, right? Right. So we're going to get into physiology today. The student version, if you wanted to use the student version, you'll need to make a copy of it. Otherwise, here we go. Moving my camera to somewhere where I'm not in the way. So physiology, hopefully you guys will find a lot of similarity between what we're going to talk about now and the sliding filament theory, because if you remember, that started out with the release of a neurotransmitter causing an action potential. And what we are going to talk about in this chapter is the signal that we send when we send nerve impulses is an action potential. So you should recognize that. So it's an electrochemical event, meaning there is a positive negative switch. That's that depolarization we mentioned with the last chapter, okay? Initiated by several different stimuli. It could be a chemo. You have chemoreceptors in your tongue and in your nose, and baroreceptors in your vessels, and proprioceptors in your muscles, and cutaneous receptors in your skin, sound mechanoreceptors in our ears, photoreceptors in our eyes. So lots of different things will cause an impulse, but when they cause it, they're causing a depolarization. So ultimately, that's what starts a signal. Okay. So a change in the neuron's plasma membrane, the neural lemma, like the sarcolemma of the muscles, but neural lemma for muscle or for nerve cells. The plasma membrane becomes permeable to sodium, same as what happened with the muscle cells to initiate the contractions. Okay. So that is depolarization. Polar means two things are opposite. So they have very different charges originally. Inside is very negative compared to the outside, very positive. As sodium comes in, the inside becomes positive and they become less different, right? So, so that's depolarizing. So as sodium rushes in, we're going to send signals from our dendrites, the receptors, up a sensory neuron through the cell body, the soma, to an associative nerve or an internerve. This is entirely within the central nervous system. Takes it to the brain, decides what to do, comes back. Sends that information off to a motor nerve, which goes out to a smooth muscle gland or organ to carry out a response. Okay? So all of this signal is being sent because of a positive negative switch on the membrane, on the neural lemma. So this is irritability. We used that word last chapter as well, the ability to respond to a stimuli. So we initiate that stimulus with a variety of things like sound, chemicals, pressure, acidity, things like that. And then that signal becomes a action potential, which is then the next stimulus, so that continues. Conductivity, the ability to transmit that impulse from one neuron to another or from one neuron to muscles or other glands. Okay, so we talked about this in the last chapter at our neuromuscular junction, where the nerve came and met the muscle. Some review of irritability. So at rest, outside of our cell, we have more sodium than inside. And inside our cell, we have more potassium than outside. That is a test question. It shows up, and I use the word cation because these are positively charged. So it's like, what is the major cation of the extracellular fluid, or what is the major cation of the intracellular fluid? So it's referring to the potassium or the sodium, and typically you find potassium inside and sodium outside, right? So we need to know the resting state if we're going to be able to talk about the switch. We use ATP after sodium has diffused out to kick it back, or diffused in, sorry, to kick it back out. Potassium will have diffused out and we have to kick it back in. So that requires the sodium potassium pump. So when the sodium rushes in, there's an influx of sodium, you would say, that inside becomes more positive and now it's depolarized. Along the axon, that depolarization causes the next sodium channel to open. So it's a gated channel and it lets sodium in, which causes the next channel to open, which allows more sodium in. So you remember that we talked about the domino effect when we did the sliding filament theory. So it's going to respond all the way or not at all. Repolarization happens after the sodium has rushed in, potassium goes out. That's repolarizing, it's reestablishing the difference in charge, okay? The problem is we don't want sodium in our cell, we don't want potassium out of our cell. So we have to use the sodium potassium pump to reestablish the original concentration gradient. I should write like sodium and potassium on my hands. So this part here has nothing to do with the charge itself. It's not depolarizing, it's not repolarizing. It's resetting the system so it can send another signal. So it's just putting it back to the initial position. When the sodium potassium pump is active, 3 sodium move out, Na plus, OUT, 3 sodium go out, 2 K plus, IN, 2 potassium go in. That's a little mnemonic you can use. So this is showing you, you have a synapse, you have a vesicle that contains some sort of neurotransmitter. Today, you're going to read about some neurotransmitters like acetylcholine, we're pretty familiar with from muscles. Serotonin, melatonin, you've heard of those related to like melatonin related to your sleep cycle, serotonin related to your kind of mood stability, right? Those are neurotransmitters. So when there's an action potential, that sodium influx change in polarity allows that vesicle to move down the axon, okay? It only goes in one direction. So when it gets to the end, like here, this is where we picked up with this, with the sliding filament theory. Remember, calcium diffused in, which allowed these vesicles to make their way to the terminal, which released through exocytosis, a biology word, our neurotransmitters into the synaptic cleft. This next cell could be a muscle cell, it could be a gland, it could be another nerve cell. No matter what, that cell has special receptors on its surface, like the acetylcholine receptor. Or, come back. The phone took over my screen. Here you are. So depending on the cell, it'll have different receptors that bind with different neurotransmitters, okay? So they'll cause different responses. So that will get sent the full length of a nerve cell. So a nerve itself is several neurons, that's your nerve cell, bound together like a cord. So a nerve is not a single cell. It's a bunch of cells all wound together. So that would be a nerve. But that one nerve may run all the way from your spinal cord to your fingers. So they're very long cells, right? So the action potential moves in one direction down the full length of that nerve. and then releases its neurotransmitter at the terminal that causes a response. So this is a close-up of that. Remember the word action potential. So from now on, when we say a nerve impulse, we mean an action potential. The ability to do work, like the potential to do action. You could think of it that way. So here, calcium rushes in. This is your synaptic bulb. The vesicles make their way to the surface and release whatever neurotransmitter they have. In this case, it's acetylcholine. Maybe it's dopamine in your brain. Makes you feel good. Okay, so that binds to a receptor at the postsynaptic cell. So then we have a series of cells all connecting together, right? And the neurotransmitter has to diffuse across a synapse. So remember, we talked about the synapse in the last chapter. And we talked about how like temperature affects the movement of molecules. So if it's warm, molecules are going to go across that synapse more quickly. And if it's cold, molecules more slow. And so they don't cross the gap as quickly. So some things can affect the speed or the ability to send that signal. And basically, it's going to have to do with those sodium channels or the calcium channels. If they can open or not open, like if they can't open, you can't send a signal, right? And that's what's going to happen with alcohol, sedatives, and anesthetics. So they reduce your permeability to sodium, or the cell's permeability to sodium. So that's why your reflexes are a little bit slower under alcohol or sedation. Anesthetics, like you don't feel pain because they're stopping your receptors from having an action potential. So the pain would be there if only the signal was being sent to your brain. Cold and pressure, I told you about cold. That has to do with the speed of the chemicals going across the synapse. Pressure, like if you're sitting too long, you might notice your foot falls asleep, and that's simply closing that neuron, which means that it's closing those channels. So if you can't have that action potential, you can't send a signal. That's all there is to that. Any questions so far? So that really should have been just a review of action potential. This part gets into the communication between cells, like what cells are talking, like which cells are talking to which cells. So when we talk about a reflex arc, the very simplest of reflex arcs is a sensory associative motor nerve, 3 cells talking to each other, okay? So it's a neural pathway. When you say reflex, you're saying that it happens without pre-thought. So it's the simplest of reactions. We have reflexes that occur inside of our body, like you have gag reflexes, right? You have reflexes that are very noticeable when you hit your leg and you when you hit your knee and you kick your leg, for example. I was telling the class earlier today, 2 days ago, I was working on my greenhouse and I have sticks in my plants holding like the leaves up. And I bent over, and without even realizing it, I poked myself in the eye with one of the sticks. And my eye like closed before I hit it. I wasn't even aware of the fact that I was going towards the stick, but somehow my eye communicated and my eyelid closed to protect my eye. And that's a reflex. Okay, so it definitely did not have thought because I didn't even know I was going towards the stick. So no integration is needed from the brain when you have a reflex. This is your chain of events. You have your sensory neuron or afferent. And when this is the one that's going to send the initial signal. So it has receptors. Its dendrites are receptors. And they're going to depolarize under certain conditions. So like on the tip of your tongue, you have dendrites that will respond to sweet chemicals. So then it's going to send a signal down. And when I say that, it's an action potential, right? Propagating down to the interneuron, which is also called the associative. So 2 names. And then that's going to relay the information to the motor nerve. So the motor nerve then is going to take it out and cause a response somewhere in your body. Your brain, like when I put my eyeball into a stick and I didn't know it until after I had already jerked my head away from it. Your brain isn't aware of a reflex until after it's already happened. So that's kind of interesting. So here's how that would look. The knee-jerk reaction is a really good example. It's because you guys are all familiar with it. We hit just below, and you can try this on your own, like just below your tibia, you have that tendon right there. And your tendons have proprioceptors, and they're responding to changes in tension, which is kind of how you know if you're like when you stand on one foot and you know you go like this to try to keep stable. That's a proprioception. So when you hit that, a sensory nerve is depolarized that sends a signal to the spinal cord. Okay. Now, something you want to notice, sensory cells. The cell body is outside of the central nervous system. Its dendrites extend into it. The associative nerve, the green one, is entirely within the central nervous system. The red is your motor nerve. Its cell body, that's the circle, its cell body is in the central nervous system, but the rest extend out. So the axon extends out of the central nervous system. So I do ask you questions related to locations. You want to be familiar with that. So in this case, the signal is sent and goes directly to the quadriceps, and you kick. And on the back route, the signal is going to go through an associative nerve for integration and coordination. And that's going to go to your hamstring to pull your leg back after you've kicked. Somatic reflexes, those are those that relate to your skeletal muscles, right? So when my eye slammed shut and my head jerked back, that was all somatic. It's skeletal muscles. So even though you don't think about doing it, it's not autonomic, right? It's skeletal muscles that those nerves are talking to. So it's somatic. So the knee-jerk reaction, the closing of the eyelid, those are somatic. Something that you can talk about here, when we were talking about synapses and how neurotransmitters have to cross that gap, something that happens when we do repetition of a homework, repetition of concepts as far as learning goes, repetition of skills like shooting a basket or a volleyball or whatever. So the dendrites grow and they get closer to the axons. So this happens through repetition, and this is your learning process, making that synapse smaller. So now my neurotransmitters don't have to cross the gap quite as far. It's going to take a lot more time for the signals to communicate, to cross the synapse, if the dendrite and the axon are far apart. Through repetition, the dendrites grow and the synapse gets smaller, and your reflex gets quicker and your recall gets quicker because you're able to just cross the gap more quickly. So that's something you're going to look at today in your lab. Autonomic reflexes have to do with things that are serviced by your autonomic nervous system, either parasympathetic or sympathetic. Okay, so a reflex does not necessarily mean automatic or autonomic, right? They do happen without thought, but that's not necessarily autonomic. These are. So like your pupil reflex, you've seen your eyes get big when it's dark and small when it's bright, right? We're protecting our retina. But we also talked about it with the sympathetic nervous system, fight or flight. Take in as much information as possible because you need to run away from a bear. Or it gets small when you're like about to fall asleep because you're so lethargic from your turkey. So this would be an autonomic reflex because these ciliary bodies that are in our eyes are a smooth muscle. Smooth muscles are serviced by autonomic nerves. The salivary reflex, like when you get digestive juices flowing, when you smell something that's wonderful, or when you bite on something that's kind of sour, you know, you feel it way back there. That's a salivary reflex. Glands are serviced by autonomic cells. So that would qualify as an autonomic reflex. Okay. So I need a follow-up slide at the end to always like bring it back together. I need to go through, and now that we're digital, I definitely need that. So from this, we just kind of reviewed the action potential, tried to really emphasize the role of sodium and conductivity and the order with which the impulse travels, sensory, associative, motor. And then we classified 2 different types of reflexes, okay? You should be able to talk through this reflex, and you're going to do that on today's worksheet as well, okay? So what questions do you have from today's topic? Let me know I'm not alone in this world. I do like when you guys are signed on to the same PowerPoint as me, because then I know that you're here too. Okay, so today's worksheet is similar to the other ones you've done. It'll relate to today's topic, and then you're going to have a little bit of fun with reflexes. Simple, very simple. So neuron physiology and neurotransmitters is our worksheet. You're going to need a time card, which you can use a ruler, or you could use a piece of paper or a note card, anything. But this is a conversion you can use. Approximately 2 inches on your card is going to be 0.1 seconds. So you're going to make a card with these divisions, or the other option, if you can print something, click this link, and then you will have time cards already printed, and you can just print that out. So then you're going to hold that. You need a friend for this, or a family member. Okay, you'll hold your fingers just at the end here, and then your friend will drop it, and then you catch it. So that's where you need the dividers, so you know how long it took you to catch that. And you're going to do it with your eyes open, and you're going to do it with your eyes closed, where they tell you to go under 3 different conditions. So you're going to see which is a faster reflex: your touch, your sound, or your vision. Okay, so so that is the second part of today's worksheet. The first part is just follow-up questions compared to today's information. Neurotransmitters, you don't really have to do anything with them. So that should look familiar. This is really just reading and getting familiar with these things for the fun of it. They're interesting. I didn't ask you any questions about it. So 1 page of questions, 1 page of information, and then this is your lab. So it's a very short lab. Do it over and over again so that you can take an average rather than an individual. And then just a couple follow-ups. So I think it'll be pretty short. You turned in your quiz and your Wiser Me today, so your attendance is all taken care of.